Today we're going to talk about DNA replication. DNA replication happens when your cells are getting ready to divide and make two new cells, and both cells need a copy of your genetic information or your DNA, and so your cell needs to replicate the DNA so that each new cell gets its own copy. So replication is the same thing as copying. A single strand of the DNA serves as a template for the new strand. The rules of base pairing are present, so A goes with T, C goes with G. Those rules are in effect for this, and the DNA is actually replicated in the synthesis stage of the cell cycle. So if you remember way back to biology, this was the cell cycle. You had G1 where it was growing, and then the synthesis stage, that is where the DNA is replicated, and then you have a little bit more growing in G2, and then you have the process of mitosis. So each body cell gets a complete identical set of DNA every time cells divide. So here's the process. So in your notes you're going to want to draw this, and you're going to want to label any parts that you think that you need to. Step 1 is the unzipping of the DNA double helix by a molecule called helicase. So let's see. This is the helicase right here. Okay, and there's one on each side, and so you've got your double-stranded DNA. The helicase kind of comes in, bumps its way in, and goes through and unzips it, going both directions. So you've got one strand of it up here at the top, and then you've got another strand down here. These two strands are going to serve as what we call a template strand, and it's going to help make the new strand of DNA. So when you draw it, you want to draw it so it's open, and then these right here are the nucleotides, right? So this is like an A, and then a T, and then whatever, and then down here would be the opposite. So if that was an A, this would be a T, so on and so forth. Okay, and they're open so that the base pairing rules can come into effect. So step 2 are free-floating nucleotides. So you've got all these nucleotides out here, A's, T's, C's, and G's, just kind of hanging out. They come in, okay, and they bond with this open strand right here. So like, for example, if this right here was an A, then this T would come in and bond right here, okay? And then if this was a C, then the G would come in, and so on and so forth. So it goes in, and the nucleotides come in, okay, and they form hydrogen bonds with the template strand. Then this big, huge molecule right here, which is step 3, called DNA polymerase, goes in after. So after those nucleotides have come in, then the DNA polymerase goes through, and it basically bonds everything together. So it adds the sugar and the phosphate, and it bonds everything together and makes the backbone. So it's starting to look like a whole molecule of DNA right here. You've got your original template strand, and then you've got your new strand, and now you've got a double helix shape again. So step 4 then, the nucleotides have formed the hydrogen bonds with the new strand, and everything kind of twists back up into the double helix shape, and now you have 2 molecules of DNA that are exactly the same, and each molecule has one old strand and one new strand, okay, twisted together to form that double helix. And this one down here has part old and part new as well. So you have 2 completely identical molecules of DNA, each one with an old and a new strand. So here's kind of the basic steps of DNA again, just so that you have them. Okay, the enzymes unzip the double helix. In this case, it's helicase. The free-floating nucleotides form hydrogen bonds with the template strand. Your A's, T's, C's, and G's come in. Then the DNA polymerase enzyme bonds the nucleotides together to form the double helix. And then the polymerase enzymes form covalent bonds between the nucleotides in the new strand. So everything kind of gets blended together. So feel free to make sure that you add these descriptions to your pictures in your notes. Here's an example of what it actually looks like.
Replication is the process by which DNA is copied during the cell cycle. Each strand of the double helix serves as a template from which a new strand is built. Click Start to zoom in and see replication in detail.
So this is your original DNA molecule that needs to be replicated.
Enzymes unzip the double helix, exposing the nucleotide bases on each strand and forming a replication bubble. These enzymes unzip the double helix at many places to speed up the replication process. This model, however, shows only one replication bubble. Additional proteins hold each strand apart so replication can occur.
So the brown enzymes that came through and opened it up, those were the helicase molecules. And now you have your two template strands right here. So this is going to be a template strand and this is going to be a template strand. And then here are all your free-floating nucleotides, and you're going to see in the next step that they're going to come in, and then right behind that the DNA polymerase is going to go through.
Each existing strand of the DNA molecule is a template for a new strand. Nucleotides pair up with the exposed bases on each strand. DNA polymerases bond these nucleotides together to form the new strands. DNA replication is smooth and continuous on one DNA strand. On the other strand, the DNA is made as a series of small fragments that are then joined together.
So as you can see, the free-floating nucleotides came in and then the DNA polymerase went through.
Replication results in two identical double-stranded DNA molecules. DNA replication is semi-conservative. That is, each DNA molecule contains one original strand and one new strand.
So a couple more important things that you can kind of add to your notes about DNA replication. It's fast and accurate. They can do all of your DNA in less than an hour's time. The DNA itself only serves as a template. Okay, it only serves as a template to make the new strand. Enzymes and other proteins are what actually do the work. Sometimes it'll be referred to as what we call semi-conservative. Semi-conservative just means there's one old and one new strand in each new molecule. Two new molecules of DNA are formed, each with an original and a new. That's what the semi-conservative means. And then DNA replication, the way that it's fast and accurate is because it starts at multiple places in a cell and goes through and not just at one point. The DNA polymerase's other job is to find and correct errors. So if it does happen to, an A comes in and bonds with a C and when it's not supposed to, the DNA polymerase will see that, detach it, and then make sure the correct one gets in. So please make sure that your notes are completely filled out, and when you're finished watching this video, you can take your notes and use it to complete the DNA replication quiz.